Alright, so today we are going to begin on our first lecture of this new unit on muscle fitness. As you guys can tell, it's a very large packet. It's just about as large, or might be even larger than the packet that we did just last week on the endocrine system. For that reason, I'm going to do the same thing I did with the endocrine system and pull 40 questions from this packet. So there will be 40 questions that come from this singular lecture, 20 come from our next lecture, which begins the cardiovascular system, and it's on blood. So your next exam, which hopefully will be next Friday, will be 60 questions. Okay. Today we're going to do a little bit of review if you've taken anatomy, or some of you are actually currently taking anatomy. These first 5 or 6 slides are going to be just straight up copy and paste, word for word, exact same stuff I cover in that class. So we're going to be able to kind of buzz through that pretty quickly, and then we'll get into the actual stuff related to muscle contractions and all that wonderful stuff that I really enjoy teaching because I enjoy muscular physiology. As you guys are all aware, since everybody in here has taken anatomy with me or is currently taking anatomy with me, there are 3 types of muscle tissue in our body. Okay. What we have pictured here on the front page is a fellow flexing. Okay, you might not recognize that fellow, but that was actually me a couple years ago before I slimmed down a little bit. Showing off his skeletal muscle. Okay? Skeletal muscle is generally what people think of when they think of muscle. Okay, but you've also got cardiac muscle, which is found in our heart. And we spend an entire lecture talking about the heart, so we'll talk more about cardiac muscle in our next unit, so that will be after spring break. And then there's also smooth muscle, which we find in quite a few different places. Those 3 types of muscle work in similar ways, which by that I mean they contract and they relax, okay, or they will sustain contractions in order to perform these 4 major functions. Okay. Now, these 4 major functions might point more towards one of those 3 than all 3. For example, the first 2 functions, the production of body movements and the stabilization of body positions, strictly relates to skeletal muscle. Okay. Our muscle in our heart and our muscle in our organs is not going to help produce body movements. They're not going to help move us. Our skeletal muscle does that. Okay. Same thing for stabilizing body position, like keeping your head upright against the force of gravity involving muscles in your neck. That's strictly skeletal muscle. So those other 2 types of muscle really relate to the 3rd function, which is moving stuff inside of us. And for example, the heart muscle, which we call cardiac muscle, that pumps blood out of the heart, obviously involves cardiac muscle. And then the smooth muscle that we find in a lot of those various organs, like our cardiovascular system, which are the blood vessels that move blood. We've also got smooth muscle throughout pretty much the entirety of our GI tract, our respiratory tract, our reproductive, our urinary tracts, our lymphatic tracts, that move those various substances. The 4th function, the generation of heat, is actually something that we've talked about since the beginning of the semester, when we talked about the negative feedback loop of body temperature regulation. As in, like when we shiver on a day like today and yesterday when it's nice and cold outside. Okay? Because as we shiver or as we contract muscles produce a very large amount of heat as a byproduct. It has everything to do with chemical reactions, because as we've learned earlier in the semester, anytime a chemical reaction occurs, heat is given off as a byproduct. Well, as our muscles contract, there's a lot of chemical reactions taking place. And that's stuff that we're going to get into not very much today, but really into on Friday. Okay. We take advantage of our shivering when we're cold to produce heat, which the actual production of heat by contracting muscles is called thermogenesis, where that word thermogenesis literally translates thermo meaning heat, genesis meaning formation, so it translates literally to heat formation or production. Although bones provide leverage and form the framework of the body, they cannot move body parts by themselves. Motion results from alternating contraction and relaxation of muscles, which make up about 40 to 50% of our total adult body weight. There we're really just talking about skeletal muscle, which is what we'll basically be talking about for the majority of this lecture. But all three types share the same primary function, which is to convert chemical energy in the form of ATP, which we get from food, into mechanical energy. That would be motion. Okay, so that allows us to perform work, generate force, and even, as it states, to produce movement. Now, those three muscle types share these four properties that we're going to get into in just a second. But first, let's talk about some similarities and differences that they have. Okay, this is straight word for word what I'd go through in anatomy. Okay, so we'll just do it real quickly here. We've learned previously that our somatic nervous system, which we just got done talking about fairly recently, is the division of the nervous system that controls our skeletal muscle and does so in a voluntary manner. Our skeletal muscle gets its name because it moves our skeleton, or the bones that make up our skeleton. A key feature of skeletal muscle tissue is that it is striated, and we're going to find out why that is today. Striations are these orderly light and dark lines that you can see in the figure on the right, which occur due to overlap of filaments within sarcomeres. Skeletal muscle attaches to bone, skin, fascia, and other muscles. It is multinucleated, and you can see several nuclei over here on the right. Each of these little kind of dark lines would be an individual nucleus. Each muscle fiber, which a fiber is a fancy way to describe a muscle cell, contains many of them, and we'll talk about why that is in just a little bit. The nucleus is going to be pressed up, or the nuclei would be pressed up against the cell membrane. So this would be a muscle cell. The nuclei will be kind of pressed up the side of it. Number 2 here will be showing another cell. Nuclei pressed up against its plasma membrane. Here will be another cell with the nuclei pressed up against it. It's the only one of our muscle tissues that's multinucleated. Cardiac muscle and smooth muscle are both uninucleated, meaning there's only one nucleus per cell. Cardiac muscle is similar to skeletal muscle in that it is striated as well, okay, because it has those light and dark orderly repeating bands. Okay. It's not as prominent in this image, but they are there. Okay. We're going to talk about skeletal muscle or cardiac muscle in much greater detail when we get to the heart. Okay. It's actually the thickest layer of the heart, what we call the myocardium. And we'll talk about the structures within the myocardium that help it to function the way that it does. The cells are really cool. They are forked or branched in many cases. And at the end of the forks, they're going to have what we call intercalated discs. These are only found in cardiac muscle. Okay? Intercalated discs are thickenings of the plasma membrane. And inside of them, and we've talked about these just a little bit in this class, are gap junctions, which are little doorways that allow ions to flow from one cell directly into a neighboring cell, which is very cool because that allows action potentials to spread from cell to cell, enabling those muscle fibers to become excited at the same time and then contract at the same time. So something that's very important to the heart because that's what allows the heart to function the way that it does. Speaking of action potentials are actually generated within the heart muscle itself in a series of cells that make up our natural pacemaker, what we call the SA or sinoatrial node, that spreads that signal throughout the heart like a wave. We've also learned previously that things like hormones and certain neurotransmitters like norepinephrine can change the rhythm of our heart. Okay. We know the sympathetic nervous system, for example, is going to do what to the speed of our heart? Speed it up. Okay. And that's because it influences that pacemaker that we have. And we'll get to that in a couple weeks. Now we know the sympathetic and parasympathetic nervous systems are part of that autonomic nervous system, which happens to be the division of the nervous system that also regulates the cardiac muscle. Autonomic nervous system also innervates our smooth muscle, which gets its name because it does not have those striations. Okay. We'll kind of talk about this a little bit less than the other two. We'll briefly mention it when we talk about things like our blood vessels and our stomach and intestines, but we don't really get into it too much. Okay. Smooth muscle is uninucleated, just like cardiac muscle. The nucleus is kind of located more within these cells than the other two. Something that's really neat about these guys is that they are slow moving generally, but fatigue resistant, okay? Which means that they don't tire out, which is something that we've all experienced with regards to our skeletal muscle. Okay? And smooth muscle is generally going to be involuntarily controlled and regulated by, as I said, the autonomic nervous system. Okay. Those three types of muscle collectively are our three muscle tissues. And those three muscle tissues share four special properties. These properties are not necessarily unique to those muscles. We, for example, talked about excitability already this semester, which is something that our neurons exhibited. So let me write, or let you guys write, and then I'll talk. Okay, we learned previously that neurons demonstrated excitability. This property is the same in muscle tissue. Once again, excitability refers to the ability of neurons in the now muscle tissue to respond to a stimulus or stimuli, which then enables them to produce electrical signals, which we call action potentials. Okay. We've talked previously about signals triggering action potentials. Okay. In muscles, there's only two types, whereas in neurons, there could have been a lot more. Okay. There's autorhythmic signals, which we'll talk about in our next unit when we get to the heart, because that's what we're talking about when we talk about the pacemaker. In this unit, we're going to talk about chemical stimuli, specifically neurotransmitters, which are released by those neurons that we've talked about. If I had followed the textbook in the same order that it runs, it talks about muscle before it talks about the nervous system. Why? Because it has everything to do with anatomy. Okay, it runs right from bones into muscles because muscles attach to bones. You need to learn all about all those processes and fossils and all that wonderful stuff right before you start talking about muscles. Just the way that I go through when I teach anatomy class. Okay. Well, in phys, it makes more sense, in my opinion, to talk about neurons and the nervous system before we get into muscles. Because now we understand what excitability is, what action potentials are, and we know what happens when an action potential reaches a synaptic end node, which causes neurotransmitters like acetylcholine to be released. That really helps us today and on Friday when we talk about action potentials and neurons. So we already know about it. It's the same essential process that occurs in muscles. Okay. Now, other chemical stimuli that can trigger muscles to contract would be hormones distributed in the blood, and we've talked about that before, or even local changes in pH. The second property is a unique property to muscle. Okay. And what it is literally right there in the word. Contractility refers to the ability to contract. Okay, contract forcefully when stimulated. Okay, so when an action potential is generated in muscles, it's going to contract. Okay. How strong it contracts is important because it must generate enough force against the resistance in order to have movement occur. And that's really just stating physics where effort or force must be greater than resistance. Okay. If I'm going to lift up a book, I must create more force than the weight of that book. Okay. The last two properties, extensibility and elasticity, are not unique to muscle. We'll see these again. And you can think of these like a rubber band. Okay. Extensibility, think extend. Another word for extend would be lengthen, which refers to the ability of muscles, and this is all three types of muscles. We've all stretched our skeletal muscles, but your heart and your smooth muscles stretch as well. And they all do so within limits without being damaged. Okay. Now, just like a rubber band, if you stretch it and let go, it's going to snap back. Our muscles will do that. And that's due to the property of elasticity. Okay. Now, muscles can also shorten and then snap back, which also relates to elasticity. We'll talk about extensibility and elasticity again when we talk about the lungs, because your lungs exhibit these properties, at least in a healthy lung. Okay. Unfortunately, smoking can cause things like emphysema, which really decrease elasticity of the lungs. And so the lungs like to stretch, they don't like to recoil. And that's why people with emphysema look like they have real big chests, because their lungs are overinflated all the time. We'll talk about that in a couple of weeks. All right, the remainder of this lecture is all about skeletal muscles. So I just kind of presented a little bit about those other muscles, cardiac and smooth. Really, the rest of this lecture and the majority of this lecture is all about skeletal muscle. Skeletal muscle is made up of cells, just like every other organ in our body, because muscles are technically organs. Now, instead of calling them cells, we like to call them fibers, which relates to their shape. Okay? And that shape that muscle fibers exhibit is kind of like a PVC pipe or a tube that you find in all sorts of places in the world. Okay? In your home as part of your plumbing, or in the ground to allow water to flow through. It's a cylinder, circular on the end, long and straight throughout the kind of three-dimensional sense. On the next page, we can see what I'm talking about, where we can zoom in at one singular muscle fiber. Now, muscle fibers are the most important component of a muscle. They are what shorten to cause a muscle to contract. And they develop early in fetal life from stem cells. And we can see their development occurring here in these kind of stages. The stem cells that begin their formation are called myoblasts. And they fuse together. As they fuse together, the nuclei are still left intact, which explains why skeletal muscle ends up as a multinucleated cell. Now, the nucleus or the nuclei will always be kind of more prominent towards the outside of our muscle fiber, just deep to its plasma membrane, which the plasma membrane is known as the sarcolemma. And I'll talk more about here in just a second. What's pretty amazing about skeletal muscles is the number of cells or fibers that make them up. At least we believe this to be the case. It's set before we're born. So in order for muscles to grow in size, which you guys would all agree with me, from a baby to a full-grown adult, muscles have to grow in size. Okay? The cells have to become wider. They don't go through cell division, which is like how your bones grow. Muscles grow by way of the cells ballooning out. Okay? A concept or process we call hypertrophy. Okay? Now, this is stimulated by a hormone that we've recently talked about. Growth hormone, as we know, obviously promotes growth, but also testosterone, which we know the men in this room produce by way of their testes, and explains why men typically are going to be more muscular than females. Now, the sarcolemma of a muscle fiber is the fancy way to describe the plasma membrane. So we come up with these extra fancy names to describe the same things we've already learned about. And it's very similar to the plasma membrane of neurons and of other cells in that it maintains a resting membrane potential. Why I said it's similar to neurons is because that resting membrane potential can change. What changed the resting membrane potential of neurons? It had to do with the flow of ions. Okay. And if enough ions, for example, like sodium, rush into a neuron, what happens to that resting membrane potential? It depolarizes, and if we depolarize enough, we can create an action potential, right? The same thing happens in muscle fibers. When an action potential is generated in a muscle fiber, it's going to spread throughout the outside of the cell along that sarcolemma. Okay? But within the muscle fiber are the actual organelles that do the contracting. These things called myofibrils, okay, which are what these cylindrical structures are in red. Okay? And we've pulled one out to show what it looks like. Okay? These myofibrils are the actual structures that contract, therefore contracting the entire muscle fiber, thereby contracting and shortening the entire muscle. To get them to contract, we need to first release calcium. And in order for calcium to be released, we have to transmit that action potential from the outside of the cell to the inside of the cell. Okay. And I kind of drew it up here last hour to show you kind of what I was talking about, or what I'm talking about. If an action potential is spreading on the outside of the muscle fiber, it's really not going to do any good. We need to take it and move it inward. Thankfully, we have these extensions or invaginations called T-tubes or transverse tubules that can carry that nerve signal inward. See these little blue tubes? They carry action potentials inward to stimulate these goldish colored organelles, which are these membranous sacs called the sarcoplasmic reticulum, which is synonymous with the smooth endoplasmic reticulum that we've talked about. The function of this organelle is to release calcium that it stores, because calcium is one of two things or two chemicals that's essential for a muscle to contract. And I'll talk about why here in just a second. Okay. So in a resting state, we store a whole bunch of calcium in the SR. Okay. If you look here, we have what's called a triad labeled, which includes a T-tube in the middle and then two terminal cisterns on the sides. The terminal cisterns are just the fat ends of the SR. Okay. Now, other things that you see within a muscle fiber, obviously, is a lot of mitochondria. Okay, that's what these kind of yellow, goldish, orangish colored organelles are. We know mitochondria are prominent organelles in cells that need a lot of energy because they produce a lot of energy. Muscle cells need a lot of energy. We'll also find a plasma, or cytoplasm, excuse me. We call it the sarcoplasm, same thing. What's not labeled there includes the stored glycogen, which we call glycosomes, or stored in glycosomes. And then a red-colored protein we call myoglobin, which is very similar to hemoglobin that you find in your blood, except it binds and releases oxygen in a way to kind of help store it in our muscles. I talk about this in anatomy, but it kind of blows people's minds a lot of times to find out that when people eat steak, that's really juicy and red. They're not actually eating it bloody. It's the myoglobin that's still in their steak that's causing the fluid that's left over to appear like blood. Okay. So, kind of a misnomer. Now, if you look at, let me go back here real quick, this bottom part of our image, where we've pulled out a singular myofibril, we see within it are these things called sarcomeres and filaments. We're going to talk about those right now. There are two major types of filaments within myofibrils. Thin filaments, which are thin, obviously, and then thick filaments, which are thick. Okay, they have the same length, they just have different widths. These are the actual structures that are involved in the contractile process. Okay. These filaments inside of our myofibrils will not actually run the entire length. If I pretend that this yardstick is a myofibril, okay, filaments would not run the entire length. Instead, they are organized at regular intervals into sarcomeres. Okay. So sarcomeres, as it states, are the basic functional units of a myofibril. And we can see that down here. They're showing one bracket. Inside of that bracket is where you find thick and thin filaments. Okay. We have thousands of sarcomeres per myofibril. Okay. And each myofibril, or each fiber, has over 1,000 myofibrils. So that's very significant. It's this pattern of arrangement, or this arrangement of thick and thin filaments within a sarcomere, that give skeletal and cardiac muscle its striated or their striated appearance. Okay. Here's what sarcomeres look like. Okay. And this is a figure very similar to the one I showed in anatomy. It's a little bit different. But we've got the borders labeled of a sarcomere, which are the Z discs, which, as it states on the previous page, are made up of dense proteins that support the thin filaments, which in the top image here are the very thin lines. Down here are the very yellow colored lines. Okay. That's what the Z discs do. Okay. At the very center of, or middle part of our sarcomere is an M line, which is made of proteins that split the thick filament in half. Okay, and then you end up with thick filaments that run this direction and part of the thick filament that run in this direction. Okay. If you look inside of the sarcomere, you'll see that there are regions where there's only thin, there's only thick, and then there's both. It's this arrangement that gives the striations. Okay. Where you only find thin filaments, which actually are split in half between two sarcomeres, are the I bands. And this is the part of the image that we saw a little bit ago that's going to look lighter in appearance. Okay. Those lighter bands. Okay. The H zone is the part of the sarcomere where there's only thick. The zone of overlap literally is named as the zone of overlap because that's where the two filaments overlap. And then there's the A band, which is going to be the darker appearing part, or the darker bands, runs from one side of the thick filament to the other side of the thick filaments. Okay. Now, when muscles contract, what ends up happening is the filaments don't change their length. Okay. What happens is the thick and thin filaments overlap each other differently. And I've got, and I'm very excited about this, a model for the first time. I just got this semester of a sarcomere to show off what happens. Okay. So here is our sarcomere. Okay, I've got these red plates on the very ends that represent my Z discs. Like on a blue shaded plate here in the middle is my M line. And then I've got these red structures that make up my thin filaments and these blue structures attached to the M line that are my thick filaments. Right now, the zone of overlap is pretty minimal between those two filaments. Okay, so this would be like a relaxed muscle. When muscles contract, the sarcomere shortens. And that is because the thin filaments get pulled to the middle of the sarcomere. Okay. And then the thick filaments get pulled closer to the Z discs. Do those filaments change their length? No, it's the overlap that changes. Now, I like this model and that top image better than the bottom image, even though the bottom image is kind of a little bit more detailed, because it shows that you have to think three-dimensionally when you think of myofibrils. They're cylindrical structures, and we kind of show that here. And if we look at the end of the image here, where we see these big red circles, they represent the thick filaments. And surrounding each one of those big red circles, at least on the inside, not the very far outer ones, will be 6 little red dots. Because every 60 degrees, there's a thin filament surrounding a thick filament. So there's 6 thin filaments that surround each thick filament. Okay. So let's talk about those filaments a little bit more and the proteins that make them up. The two major proteins that are part of a sarcomere are myosin and actin, and these happen to be classified as our contractile proteins. I'm going to skip ahead a couple slides to show you the figures here that illustrate their structure. Okay. Now, for each individual myosin, you can see in letter A, it looks like you've taken 2 golf clubs, and I talk about this in anatomy, so hopefully you guys can kind of remember this, and you twist them around each other with regard to their shaft, and then the heads are left intact. Okay. What's different about myosin, each individual head can be moved, because right here at this kind of neck region, it can swivel. Kind of like my wrist can flex and extend. Okay. That allows the heads to pull on the thin filament. Okay. And that's what causes the sarcomeres to shorten. Now each head has to grab the thin filament, and that is going to be where we see these actin binding sites, which literally are going to tell you exactly what it does. Binds to actin, which happens to be our major component of the thin filament, and I'll get to that in a second. Okay. There's also ATP binding sites, which tell you that it binds to ATP, because energy is essential for muscles to contract. Okay. Now, individually, one thick filament is made up of hundreds of these individual myosin molecules, where some run this direction towards what would be the Z disc over here, and then the other run to the Z disc on the other side. So they run both directions within our sarcomere. And you can see that over here in our images on the right. Okay. Now, making up the thin filament, we've got 3 proteins. The major protein is actin. Okay. Now, actin is structured a little bit different, quite a bit different. It is made up of beads, kind of like beads making up a necklace. Okay. We have individual beads known as G-actins that get stranded together to form what we call F-actin strands. Okay. And a singular thin filament is made up of 2 F-actin strands helix around one another. So it'd be like taking two beaded necklaces and wrapping around each other. Okay. On each one of these little beads, our G-actins, is a black dot that represents the myosin binding site. Guess what binds to each one of those? Myosin. Okay. So myosin had an actin binding site. Actin has a myosin binding site. That's where they meet and form what we call a cross bridge. And we'll talk about cross bridges on Friday. Okay. Now, in a resting state, this cannot occur. And that has to do with these two proteins that we're going to talk about right now. I'll let you guys write, and then we'll talk. Those other two proteins that make up the thin filament and were illustrated on that figure are called troponin and tropomyosin. We call these regulatory proteins because they contract or they control or regulate or switch, whatever you want to use to describe it, the contraction process. Our figure here on this next page shows at the very bottom what an intact thin filament looks like. Okay. Those tropomyosins and troponins are added to the F-actin strands to form the thin filament. This happens to be a thin filament in a resting state. And I can tell you that with 100% certainty because I can't really see those binding sites. They're covered up by the troponin or tropomyosin molecules. Okay. What happens to move those and to expose those binding sites relates to that calcium that I was talking about a little bit ago. Calcium that's released from the SR will bind to troponin and can shift the tropomyosin, exposing those binding sites. And we're going to talk about that on Friday. There's a third type of proteins that you find in the myofibrils. They help keep the thick and thin filaments in proper alignment, give the myofibril elasticity and extensibility, and then help link the myofibrils to the sarcolemma and extracellular matrix. The most prominent and the third most plentiful protein in our myofibrils and in our muscles as a whole is titin. Titin, which is a word that reflects its large size. Okay. It was actually labeled in our figure of a sarcomere a couple of slides ago. Function of it is to stabilize the position of myosin by spanning from Z discs to M lines. Okay. This is where much of that elasticity and extensibility of skeletal muscle, cardiac muscle comes from. Okay. Dystrophin is a second type of structural protein. It links thin filaments and sarcomere to integral membrane protein in the sarcolemma. You probably have heard of muscular dystrophy before. The most common form, Duchenne's muscular dystrophy, relates to a kind of invalid or incorrect form of dystrophin. It causes muscle weakness and can even kind of cause, not necessarily paralysis, but inability to contract muscles. Very debilitating disease, and really there's no cure for it. So it's a terrible, terrible disease. And then finally, nebulin is the last type of protein, structural protein, wrapping around the entire length of each thin filament, helping anchor them to the Z discs. On the right here is an illustration showing you, and I kind of already did it with my model, but demonstrating what happens when you go from a relaxed muscle to a partially and then maximally contracted state. Okay, we're showing two sarcomeres. We're really highlighting H-zone, I-band, and A-band. H-zone, once again, remember, is just the thick. I-band is just the thin. A-band is the entirety of the thick. As we move towards maximally contracted, we see the H-zone disappear because the thin filaments are moving to the center of our sarcomeres. The I-bands are disappearing for the exact same reason, except this time the thick filaments are moving towards the Z discs. And then the A-band is unchanged because those filaments don't change their length. They just slide over one another. It's something I always like to really, really illustrate and emphasize. So any questions on any of that? Most of this, like I said, has been reviewed. What we're going to do now is actually talk about muscle contraction, where the fun really starts to begin. Before a muscle can even think about contracting, we need a nerve signal. Once again, demonstrating why I like to talk about the nervous system way before we talk about muscle contraction. And as we know, since we're talking skeletal muscle, the division of the nervous system that innervates our skeletal muscle is our somatic nervous system. And therefore, we're going to be dealing with somatic motor neurons that will stimulate the muscle fibers to contract. So down here at the bottom, we're showing a figure of one singular synaptic knob or end bolt, one of those very small swollen appendages at the very tips of those axon terminals, which we know is where we store neurotransmitters. And the neurotransmitter involved in muscle contraction is acetylcholine, which we've talked a lot about so far this semester. We know acetylcholine is abbreviated capital A, capital C, capital H. And when that action potential arrives at the knob, calcium enters it, triggers the vesicles to spit the acetylcholine out into the synaptic cleft. Now, instead of another neuron here, we have the plasma membrane of the muscle. Across this cleft, we have this invaginated region of the sarcolemma, which is called the motor end plate. That's the fancy name to describe the plasma membrane that's going to receive the acetylcholine. If I go back to the figure, the reason that it's invaginated, it has these up and down regions, is to increase the number of these little purple things, which if we zoom in, we find out that each one of those little purple things is a acetylcholine receptor, which, that's exactly what it sounds like, binds with acetylcholine. And guess what happens when acetylcholine binds to them? It opens up the flow of ions. And then we can have sodium rush in, we can have potassium rush out, but more importantly is the sodium rushing in. And we've learned all about what happens to resting membrane potential when sodium rushes in, right? It becomes depolarized. And we create a local or graded potential. And if enough sodium rushes in, we hit that keyword, threshold, and therefore triggering an action potential. And that's the point when muscle fibers become excited. It's the same principle that we've seen before, which is nice, right? Especially if you still understand it. Remember, if you don't, it might not be a bad idea to go back and review it a little bit. As the sodium rushes in, it's going to trigger the opening of those voltage-gated sodium channels that we saw in our neurons, because our muscle fibers have them as well. Okay? And as we know, just like in neurons, when an action potential was generated in the trigger zone of the axon hillock, the action potential is not going to stay still, is it? It's going to spread like a wave Instead of going down the axon, what's it going to do? It's going to spread throughout the entirety of that sarcolemma. Okay. And inward by way of those T-tubes, or transverse tubules, which is what we're going to talk about on Friday. Okay, so we're going to pause here for today and pick up with what happens next. We haven't got into the actual contraction process yet, have we? All we're doing is just getting us ready for the contraction. And it involves action potentials. Because why not? Action potentials are the best.